after learning about transcription, we will move ahead with the central dogma where DNA undergoes transcription to form the RNA. Then furthermore, RNA in association with ribosomes undergoes a process called as translation to form the proteins, which are the ultimate expressions in the individuals. So in this lecture, today we will discuss about translation, the process, how it takes place, what are the machineries involved during the process. So we start with the lecture, translation. Now what is translation? Translation is the process of synthesis of protein from mRNA with the help of ribosomes. Now proteins, we are familiar, proteins or polypeptides, they are made up of amino acids. So what is translation? Translation is the process whereby amino acids will get linked with the help of peptide bonds or will undergo polymerization to form a polypeptide or protein. So that is the process of translation. Now during translation, what happens? Or ultimately, what is translation? Physically, okay, it is the formation of polypeptides. But during that formation, what is actually happening is the genetic information which is present in the nucleotides, which is present in the DNA or the genetic material of that particular organism, the genetic information that is locked within it, that is transferred to a polymer of amino acids. That is transferred in the form of a polypeptide which is made up of amino acids. Now, what directs the process of translation? The process of translation is directed by the genetic code. Now, the relationship between the sequence of amino acids in a polypeptide and the nucleotide sequence of DNA or mRNA is called as genetic code. I repeat, the relationship between the sequence of amino acid during that protein synthesis or in the polypeptide and the nucleotide sequence which is present in the DNA from which the RNA was produced, mRNA was produced, that relationship is called as the genetic code. So to say, the nucleotides which are present in the DNA, the nucleotides which make up the DNA, those nucleotides will direct the arrangement of amino acids for the formation of the polypeptide. So that coding sequences, that is referred to as the genetic code. Now, genetic code was studied for a long time by different scientists. They had studied about or tried to decipher the genetic code, how actually it happens. Many scientists, contribution of many scientists are there. The most significant of them we can say is George Gamow, who was actually a physicist. He was the person who suggested that the genetic code is made up of 3 nucleotides. That is, it is a triplet. He suggested that the code must be made up of 3 bases. The code must be made up of 3 bases in order to code for 20 amino acids. What he said was that there are only 4 bases: adenine, guanine, cytosine, uracil. If we talk of the RNA, 4 bases. Those 4 bases, if they come together or if they work in sets or groups of 3, that is 4 raised to 3. That is the equation that we get. So it will give us 64. 64 codons are possible when we take the combination of 3 bases per code. So that way he suggested that the codons are triplets. They are made up of 3 nucleotides. Later, other scientists, Hargobind Khorana, Marshall Nirenberg, Severo Oka, they all demonstrated different work on genetic code. But here we will mainly stress on the genetic code, the salient features of the genetic code, which was given out by George Gamow and other scientists. Now, it is suggested in the salient features that the codon is triplet, as I just now mentioned. Codon is triplet, that is, it is made up of 3 nucleotides. So, in that context, we get 64 combinations. But out of those 64 combinations, only 61 codes for amino acids. In nature, there exist 20 amino acids. So those 20 amino acids are actually coded by only 61 codons and not all 64. 61 codons, they code. They are the code for these 20 types of amino acids. Then what about the rest 3? Those 3 are referred to as stop codons, which do not code for any amino acid. They will help in termination of the formation of polypeptide. Furthermore, it is also stated in the salient features that one codon will always represent one amino acid. One codon cannot represent 2 different amino acids. Hence it is known as unambiguous and specific. One amino acid can be coded by more than one codon. That means 2-3 codons, because there are 61 codons coding for amino acids whose number is just 20. So we have more number of codons compared to the number of amino acids. So it was concluded, of course, through several experiments, it was concluded that one particular amino acid may be coded by more than one codon, and hence we get 61 codons. That is why we have 61 codons. Some amino acid may be coded by only one codon, but others may be coded by 2 or some may be by 3. So the genetic code is said to be degenerate. The nucleotides, they are always read in a continuous manner. It is universal from the smallest of the organism, bacteria, protozoa, fungi, algae, higher plants, angiosperms, gymnosperms, animals, humans, birds, insects, any living organism we talk of, the codon is universal. Means whatever codon codes for a particular amino acid in a bacteria, the same will be even in case of human or any other living being that we talk about. Then furthermore, the codon AUG. The codon, this is a triplet, one of the triplets out of those 61 codons. AUG is said to have a dual function. That means it has 2 functions. What are the 2 functions? Firstly, of course, it codes for amino acid methionine. Here in this case, single codon, single amino acid. So it codes for methionine, first function. Second function is that whenever translation takes place, methionine or this AUG codon will be the first codon which will start with the translation. No other codons can start with the process of translation. That is why it is also called as the initiator codon. So, AUG is said to be having a dual function or 2 functions. Now we come to transfer RNA. Now, what is transfer RNA? Transfer RNA is a molecule of ribonucleic acid whose structure is single-stranded, quite like other RNAs, but it folds upon itself. You can see here, this is a 3-dimensional view. In reality, it appears in this manner. So, it folds upon itself in such a manner that it it seems like double-stranded, but actually it is not double-stranded. The single strand itself folds upon itself, and at the end we get both the open ends together. So here we can see 5' and 3'. So this is a single-stranded structure. As you can see here, the way it is folded. I'm pointing my cursor. Here the pointer is pointed. So ultimately what happens, this is a single-stranded structure, but it gets folded upon itself. The actual model, it looks like an inverted L. We try to represent it with the two-dimensional view for making our studies easier. Now in the two-dimensional view, it looks like a clover leaf model. Here 5', 3'. Now they have some specific ends. Even within this itself, it has the specific ends. That means the ends we determine with what it is attaching. For our studies, we are more focused on two ends only. We will talk about two. That is, one end, the open side, is referred to as amino acid attachment site, to which the amino acid will attach. Why the amino acid is attaching? Because transfer RNA, its role itself is to transfer amino acid to the site of protein synthesis, that is to the ribosomes. So it is very much important that the tRNA should attach with the amino acid, should be able to carry the amino acid. So the amino acid will bind to this open end site. Now there are 20 types of tRNAs for 20 types of amino acid. That means for each amino acid there is a specific tRNA. What will determine that tRNA, or what will determine which amino acid will link to which type of tRNA? That will be determined by the anticodons. Now what are these anticodons? Anticodons are anticodon end. This is called as anticodon end, which has 3 bases which are exposed. Now these 3 bases, the others we can see they are forming temporary bonding, but these 3 bases, they do not pair up with any others. These 3 bases, they are exposed. They may be like say UAC. Suppose they are UAC. Here 3 bases are present, UAC. What is the complementary of UAC? Complementary of UAC is AUG. So complementary basis of AUC is AUG. Now AUG is the codon for methionine. So if the anticodon is AUC at this end, then what will happen? The other end, that is the amino acid end, upon it will bind methionine, because it has the anticodons UAC, the anticodon for the codons representing methionine. Likewise, so this is also a simplified view of the same tRNA. It's just the diagram is shown in a different way. So suppose here also the anticodons, let us assume another example I'll give here. Suppose the anticodons are, that means at this end the codons present are AAA, which we are calling as the anticodon. So AAA, what is the complementary of AAA? U. These are the complementary bases of UUU are the complementary bases of AAA. Now UUU, it codes for phenylalanine. So what will happen? At the amino acid end, there will be binding of the amino acid phenylalanine. It will bind. So what type of amino acid will bind at the attachment site will be determined by the anticodons that are present at the anticodon end. So that is how the tRNA will help in carrying of amino acid. So what is tRNA? It is an adapter molecule because it helps in transfer of amino acids during translation. It has an anticodon loop. It has an amino acid acceptor end. It looks like the secondary structure looks like the cloverleaf model. Actual structure looks like an inverted L. Now we move over to the process of translation. Now the process of translation, what are the machineries involved? The main machineries involved are mRNA. First, ATP, tRNA, ribosomes, and the catalyst. That is, the catalysts are like say ribozyme or peptidyl. These are the machineries involved in the process of translation. Now, what does the mRNA do? mRNA will define the sequence of amino acids which are to be lined up for the formation of the polypeptide. So mRNA will have the sequence of the nucleotides which will work in triplets. It is the full form of mRNA is messenger RNA. So what does it do? It brings coded information from the DNA, and after that it takes part in translation and will help in bringing the amino acids in the particular sequence during the formation or synthesis of polypeptide. Okay. Now, ATP, it is required for energy, as we are quite familiar. Transfer RNA, what is it required for? Transfer RNA is required for the transfer of amino acid from the amino acid pool. Now, what is the amino acid pool? Amino acid pool is the cytoplasm. Within the cytoplasm, there is deposit of amino acids. So those from that pool, tRNA will help in transferring amino acid to near the mRNA where they will line up. Then ribosome. Now, the ribosomes is the cellular factory. Okay, it has its own ribosomal RNA and it acts as the cellular factory. So, protein synthesis occurs over the ribosomes, upon the ribosomes. Each ribosome has two unequal parts, small and large. The larger subunit of ribosome and the newly formed polypeptide, they remain attached. The smaller unit of ribosome helps in attachment of the mRNA and then starts with the translation process, which we will be seeing in the coming slides. Then the enzyme ribosome will help in peptide bond formation, that is linking the amino acids to form the polypeptide. Finally, a release factor which will help in release of the polypeptide from the tRNA. Release factor from the tRNA and from the mRNA. Now we go to the translation unit. What makes up the translation unit? The translation unit is made up of, that means the translation unit is a part of the ribosomal, it is a part of the messenger RNA which is attached upon the ribosome. It will consist of, or it has to consist of, a start codon. It has to have a codon, the nucleotides in the sequence of AUG. Following AUG, the start codon, it will have a region which will code for polypeptides. And following, whatever be the length will vary. Now, whatever be the length of the region coding for polypeptide, finally there will be the stop codon. One of the stop codons which we saw in the previous slide, one of the 3 stop codons will be present there. This portion, that is from AUG till the stop codon, will form the polypeptide. Now, before the start codon and after the start codon, there will be some regions, there will be some bases which are referred to as the UTR, untranslated regions, that cannot undergo translation. So here I will just show one mRNA. Here we have an mRNA with 5 and 3 prime. Now it might have different bases. Suppose here there are some bases. We are not naming them. Few bases, and somewhere here is present our bases, that is the start codon bases AUG. Following which there are again some other bases, many bases are present here. Then again here we have, suppose, one of the stop codons. Let us take it as UA UAG. Suppose here is present the three bases UAG. Again, after that there are some bases, let us assume. So here we are not writing the bases. Now this whole structure of mRNA will be the translation unit. So what is the translation unit made up of? As told here, the start codon AUG, it should have a start codon from where will start the protein formation. This will help in protein formation. It starts, then these are the regions that will code for the amino acids, thereby forming the polypeptide till the time it reaches the stop codon. Once it reaches the stop codon, no more formation will be possible. So from this specific part, the polypeptide will be formed. But before the start codon also there were some bases, and after the stop codon also there are some bases. This will never participate in translation. Hence they are called as untranslated regions. Now there are three steps. In 3 steps the translation process takes place. They are initiation, elongation and termination. We'll see them one by one. Initiation. In initiation, first what happens is amino acylation of tRNA. That means the tRNA gets attached or comes in combination or combines with amino acid. During that step, it takes place in a sequence. First, the amino acid will bind with an ATP molecule, form an amino acid AMP enzyme complex and inorganic phosphates. The whole structure will then be linked up with the tRNA. That is amino acylation of tRNA or charging of tRNA. Next step, almost parallelly maybe, it happens. The mRNA which has just been transcribed will attach with the ribosome. Will attach with the ribosome. The ribosome, they have P and A site. The ribosomes, they have P, A and one more E site also. This is exit site, but we are not much concerned with it because it just helps in removal of the transfer RNA. So mainly we talk about P and A site. P stands for peptidyl site, A stands for amino acyl or acceptor site. So now the P site, this is upon which the transfer RNA with the amino acid will bind. So initiation. During initiation, what happens? The smaller unit of ribosome, this is the smaller unit of ribosome. To the smaller unit of ribosome, the mRNA will first bind. Followed by attachment of the tRNA carrying the initiator amino acid or with the initiator codon. So this association, this part is included within initiation. So initiation involves 2 steps. One is amino acylation of charging of tRNA, and secondly the association of mRNA upon the ribosome and binding of initiator transfer RNA upon the start codon. Furthermore, initiation little more we discuss here. So amino acid ATP binds, then the enzyme complex, these 3 forms a complex first, which then attaches with the transfer RNA, following which the enzyme complex moves out, AMP moves out, leaving behind only the amino acid bound upon the transfer RNA. Now this transfer RNA is ready to carry the amino acid to the ribosome where polypeptide formation will take place. Simultaneously, what happens? Smaller unit of ribosome upon which the messenger RNA attaches or associates. It moves over, the ribosome moves over the mRNA. It moves over the mRNA till it at a specific location. When the AUG, the start codon, reaches the specific location, at that point the signals will be sent. So accordingly what will happen? The tRNA carrying methionine, which is already present. Here we can see it has got amino acylated with methionine, with proline, with phenylalanine, with glutamic acid, with all the 20 types of amino acids. They will be present in the cytoplasm. So, but here as the codon is for methionine, so transfer RNA carrying methionine will come and bind here. It will attach here. Temporary attachment will take place here. It will get attached. As soon as it gets attached, simultaneously or parallelly, what will happen? The larger subunit will come over it, as we can see here, the larger subunit. And here, where did it get attached? It got attached over the P site, peptidyl site here. This is the initiation step. And here, before AUG, whatever region was present, all these are untranslated regions. Next is elongation. In elongation, what will happen? The P site, as we just now saw, okay, in the P site, AUG codon was present, to which was attached the transfer RNA carrying methionine, which has anticodons USC. Now, the next site, the site following P is A, amino acyl or acceptor site. This will also be, even here will be present the three bases, which will act together as triplets. Here, as you can see, the bases are CCG. CCG is the codon for proline. It is the codon for proline as per genetic code. Now, the tRNA to which is attached proline, it will have the anticodons GGC. Now, GGC will move here and get temporarily attached. Being complementary, they will get temporarily attached. Then here, as you can see, it has got attached here. Now, when the tRNA carrying proline takes position, that side already methionine was there. Now, what will happen? Methionine and proline, they will lie quite side by side. And then they will form a bond here. Of course, with the help of enzyme, the ribosome. Okay, they will form the bond. And what bond? That bond is the peptide bond they will form. The ribosome will not remain in the same position. It will keep on moving. It will keep on moving as it moves. As it moves, what will happen? Now, there will be shifting. This AUG will be out of the ribosome. CCG will be in the P position and the A position will be occupied by UAU. So, what will happen now? As AUG goes out, UAC, the tRNA with anticodon UAC will no more be able to remain bound upon the AUC because it is already out of the ribosome. So, following this, what will happen? The tRNA will move out. As it moves out, it will not carry methionine with it, which was attached. Why? Because methionine now is held by the proline, which in turn is attached here. We can see how it is linked. So, only the transfer RNA will move out. The one carrying proline with the codes, anticodes, everything will occupy the position P. A will be occupied by UAU. So, the next tRNA carrying the particular amino acid will come and bind or take position with UAU. The third amino acid comes, the third one will also bind. Following which, when the third amino acid forms the peptide bond, so that way one by one the amino acids will go on lining up, which are carried by the transfer RNAs. So in elongation phase, what happens? There is repeated cycle of amino acids which are delivered by tRNA. Peptide bond formation and movement along the mRNA takes place. Now the termination, the last step. In this, what will happen? Now, after many amino acids have lined up and formed the peptide bonds, they have been linked up with the peptide bonds. Now, what happens is, let us assume that mRNA was quite long with different codons, triplets. It reached a codon which has one of the stop codons. Let us assume UAG. So, here UAG is present. Now, when the codon just ahead of UAG has reached the P site, UAG has reached A site. So, we have to remember the sites very well. So, the preceding, the stop codon has reached P, the A site, UAG or the stop codon has reached. Now, what happens? The stop codons do not have any corresponding tRNA or corresponding amino acid. So, there is nothing which can come and bind upon these three codes, which can bind upon these three bases, this particular triplet UAG, It can be any one of these three. So there is nothing to bind upon it. So, but that position, at that point, that position will be occupied by a release factor, which is a protein. It's a type of protein. The other steps, the ribosome is continuing its movement. It keeps on moving. As it moves, what will happen? That the tRNA, the preceding one, it is already out. Once it is out, now what will happen? It will become free. It will get free from the ribosome, and then it will get free from the ribosome, and that side the amino acid chain will be released out. The amino acid chain will be released out of the ribosome. So that terminates the polypeptide formation. We can see here the polypeptide is free, its formation is complete. Following which, even the ribosome will dissociate into its smaller and larger subunit. The mRNA release factor, everything gets separated out. With that culminates or terminates the process of translation. So in termination, we have the terminator codons. One of the terminator codons, when it reaches the A site, termination will take place. No charged tRNA can bind upon these codons. The release of polypeptide will be catalyzed by the release factor. So with that, the formation of polypeptide terminates, and the protein formation which had started from the transcription process is completed. And finally, there is expression of the Mendelian characters in the form of the proteins.